MHC genes and gene products. Hello to all the listeners and viewers. The topic of discussion here is MHC genes and gene products. To learn about MHC molecule and gene, it is very much necessary to get accustomed with the term MHC. So, let us look upon first to concept of MHC. MHC is known as major histocompatibility complex, is a group of genes that code for cell surface histocompatibility antigens and are the principal determinants of tissue type and transplant compatibility. Histocompatibility antigens refer to any of the genetically determined antigens on the surface of cell membranes that serve to identify a cell as self or non-self. These antigens determine whether a tissue graft will be accepted by an organ transplant recipient. They are the most diverse genes in humans and are used to determine if a sample of DNA comes from a specific person. MHC affects the response of an individual to antigens of infectious organisms and is therefore implicated in the development of autoimmunity. The present discussion will explore briefly the basics of MHC and describe the genes and molecules encoded with it. We will start with the history. History. Gorer in the year 1930s gave two ideas. Rejection of foreign tissue is the result of an immune response to cell surface molecules. Identification of first, second, third and fourth groups of genes. Gorer and Snell in 1940s and 1950s emphasized that antigens encoded by the genes in the group 2 took part in the rejection of transplanted tumors and other tissues. Snell called these genes histocompatibility genes, currently called H2 genes. Snell was awarded the Nobel Prize in the year 1980. Let us have a look upon the general organization of MHC classes. General organization. Major classes of MHC. The MHC gene family is divided into three subgroups: class 1, class 2 and class 3. Of the three MHC classes identified, human attention commonly focuses on classes 1 and 2. By interacting with CD4, that is cluster of differentiation molecules on surfaces of helper T cells, MHC class 2 mediates establishment of specific immunity, also called acquired immunity or adaptive immunity. By interacting with CD8 molecules on surfaces of killer T cells, MHC class 1 mediates destruction of infected or malignant host cells, the aspect of specific immunity termed cellular immunity. The other arm of specific immunity is humoral immunity, whose relation to MHC is more indirect. The detailed description about each class will be discussed now. MHC class 1. MHC class 1 occurs on all nucleated cells apart from RBCs and presents epitopes to killer T cells, also called cytotoxic T lymphocytes or CTLS. A CTL expresses CD8 receptors in addition to T cell receptors, that is TCRs. When a CTL's CD8 receptor docks to MHC class 1 molecule, if the CTL's TCR fits the epitope within the MHC class 1 molecule, the CTL triggers the cell to undergo programmed cell death by apoptosis. Thus, MHC class 1 helps mediate cellular immunity, a primary means to address intracellular pathogens such as viruses and some bacteria, including bacterial L forms, bacterial genus Mycoplasma and bacterial genus rickettacea. Structure of MHC class 1 protein. MHC1 occurs as an alpha chain composed of 3 domains alpha 1, alpha 2 and alpha 3. The alpha 1 resets upon a unit of the non-MHC molecule beta 2 microglobulin and coded on human chromosome 15. The alpha 3 subunit is transmembrane, anchoring the MHC class 1 molecule to the cell membrane. The peptide being presented is held by the floor of the peptide binding groove in the central region of the alpha 1 alpha 2 heterodimer, a molecule composed of 2 non-identical subunits. The genetically encoded and expressed sequence of amino acids, the sequence of residues of the peptide binding grooves floor determines which particular peptide residues it binds. Classical MHC molecules present epitopes to the TCRs of CD8 plus T lymphocytes. Non-classical molecules, that is MHC class 1b, exhibit limited polymorphism, expression patterns and presented antigens. This group is subdivided into a group encoded with MHC loci. For example, HLA-E, HLA-F and HLA-G, as well as those not encoded, for example, stress ligands. The antigen or ligand for many of these molecules remain unknown, but they can interact with each of the CD8 plus T cells, NKT cells and NK cells. MHC class 2. MHC class 2 can be expressed by all cell types, but normally occurs only on professional antigen presenting cells, that is APCs, macrophages, B cells and especially dendritic cells or DCs. An APC uptakes an antigen, it then carries out antigen processing and returns a molecular fraction of it, which is known as epitope, to the APC's surface that is coupled within an MHC class 2 molecule. On the cell surface, the epitope can contact its cognate, that is the relative molecule, which helps in matching molecular region on immunologic structures and finally recognize that epitope. That molecular region which binds or ligates the epitope and then is finally known as is the paratope. On surfaces of helper T cells, there is presence of CD4 receptors as well as T cell receptors or TCRs. When a naive, that is newly formed helper T cells CD4 molecule binds to an APC's MHC class 2 molecule, its TCR can meet and be imprinted by the epitope coupled within the MHC class 2. This event primes the naive helper T cell. According to the balance of cytokines secreted by APCs in the microenvironment, the naive helper T cell polarizes or differentiates into either a memory Th cell or an effector Th cell of phenotype is either type 1, that is Th1, type 2, that is Th2, type 17, that is Th17 or regulatory or suppressor, that is T regulatory. MHC class 2 thus mediates immunization. The polarization during primary exposure to an antigen is key in determining chronic diseases such as inflammatory bowel diseases and asthma by changing the immune response that memory Th cells coordinate when their memory recall is triggered upon secondary exposure to similar antigens. B cells express MHC class 2 to present antigen to Th0. Structure of MHC class 2 protein. MHC class 2 is formed of 2 chains alpha and beta, each having 2 domains alpha 1 and alpha 2, beta 1 and beta 2. Each chain is having a transmembrane domain alpha 2 and beta 2 respectively, anchoring the MHC class 2 molecule to the cell membrane. The peptide binding groove is formed of the heterodimer of alpha 1 and beta 1. MHC class 2 molecules in human have 5 to 6 isotypes. Classic molecules present peptides to CD4 plus lymphocytes. Non-classic molecules accessorize with intracellular functions, are not exposed on cell membranes, but in internal membranes in lysosomes, normally loading the antigenic peptides onto classic MHC class 2 molecules. Having known about the basics of MHC proteins, let us now look upon two MHC genes. MHC genes. MHC gene families are found in all vertebrates, though they vary widely. In human, the MHC region occurs on chromosome 6 between the flanking genetic markers MOG and COL11A2 and contains 140 genes spanning 3.6 mega base pairs, that is 3.6 mega base pairs or 36 lakh bases. About half have known immune functions. Class 3 functions very differently from class 1 and class 2, but its locus occurs between the other two classes on chromosome 6 in human. The exon-intron arrangement of class 1 and class 2 genes. Separate exons encode each region of class 1 and 2 proteins. Each of the mouse and human class 1 genes has a 5' leader exon encoding a short signal peptide followed by 5 or 6 exons encoding the alpha chain of the class 1 molecule. The signal peptide serves to facilitate insertions of the alpha chain into the endoplasmic reticulum and is removed by proteolytic enzymes in the endoplasmic reticulum after translation is completed. The next 3 exons encode the extracellular alpha 1, alpha 2 and alpha 3 domains, and the following downstream exons encodes the transmembrane, that is TM regions. Finally, 1 or 2 3' terminal exons encode the cytoplasmic domains. Like class 1 MHC genes, the class 2 genes are organized into series of exons and introns mirroring the domain structure of the alpha and beta chains. Both the alpha and beta genes encoding mouse and human class 2 MHC molecule have a leader exon, an alpha 1 or beta 1, an alpha 2 or beta 2 exon, a transmembrane exon and one or more cytoplasmic exons. There is correspondence between exons and the domains in the gene products. The mRNA transcripts are spliced to remove the intron sequences. Each exon, with an exception of the leader exon, encodes a separate domain of the MHC molecule. The leader peptide is removed in post-translational reaction before the molecule is expressed on the cell surface. The gene encoding beta 2 microglobulin is located on a different chromosome. Structure. The MHC complex resides in the short arm of chromosome 6 and overall size of the MHC is approximately 3.5 million base pairs. The complete 3-dimensional structure for both class 1 and class 2 MHC molecules has been determined by X-ray crystallography. The class 1 gene complex contains 3 loci A, B and C, each of which codes of alpha chain polypeptides. The class 2 gene complex also contain at least 3 loci DP, DQ and DR. Each of these loci codes for 1 alpha and a variable number of beta chain polypeptides. Class 3 region is not actually a part of the HLA complex, but is located within the HLA region because its components are either related to the functions of HLA antigens or are under similar control mechanisms to the HLA genes. Class 3 antigens are associated with proteins in serum and other body fluids, for example, C4, C2, factor B, TNF, and have no role in graft rejection. Class 1 is encoding for peptide binding proteins which select short sequence of amino acids for antigen presentation as well as molecules aiding antigen processing such as TAP and tepecin. Class 1 expression is shown by one chain called alpha whose ligands are the CD8 receptor borne notably by cytotoxic T cells and inhibitory receptors borne by NK cells. Class 2 encoding peptide binding proteins and proteins assisting antigen loading onto MHC class 2's peptide binding proteins such as MHC2DQ, MHC2DR and MHC2DP. Class 2's expression is shown by two chains called alpha and beta whose ligands are the CD4 receptors borne by the helper T cells. Class 3's encoding is other immune proteins outside the antigen processing and presentation such as components of the complement cascade, for example, C2, C4, factor B, the cytokines of immune signaling, for example, TNF alpha, and heat shock proteins buffering cells from stresses, while class 3 expression is shown by various. Cellular distribution of MHC class 1 molecules. In general, the classical MHC class 1 molecules are expressed on most somatic cells. The highest level of class 1 molecules are expressed on lymphocytes. 1% of the total plasma membrane proteins or 5 into 10 to the power 5 molecules per cell. Fibroblast, muscle cells, hepatocytes and neural cells express very low levels of class 1 molecules. A few cell types, for example neurons and sperm cell at certain stages of differentiation appear to lack class 1 MHC molecules altogether. Cellular distribution of MHC class 2 molecules. Class 2 molecules are expressed constitutively only by antigen presenting cells that are APC, for example macrophages, dendritic cells and B cells. Thymic epithelial cells or some other cell types can be induced to function as APC and then express class 2 molecules under certain conditions. Cellular distribution of MHC class 3 molecules. Class 3 molecules are not membrane proteins, are not related structurally to class 1 and class 2 molecules and have no role in antigen presentation, although most play some role in immune responses. For example, C2, C4A, C4B, factor B, 21 hydroxylase enzymes, TNF alpha, TNF beta, heat shock proteins, that is HSP. Inheritance. Histocompatibility genes are inherited as a group that is haplotype, one from each parent. Thus, MHC genes are codominantly expressed in each individual. Each individual inherits a maximum of 2 alleles for each locus. The maximum number of class 1 MHC gene products expressed in an individual is 6, that for class 2 MHC products can exceed 6, but is also limited. Thus, and each chromosome is found twice, that is diploid in each individual, a normal tissue type of an individual will involve 12 HLA antigens. Haplotypes normally are inherited intact and hence antigens encoded by different loci are inherited together. However, on occasions there is crossing over between two parental chromosomes, thereby resulting in new recombinant haplotypes. There is no somatic DNA recombination that occurs for antibodies and for the TCR. So, the MHC genes lack recombinational mechanisms for generating diversity. Many alleles of each locus permit thousands of possible assortments. There are at least 1000 officially recognized HLA alleles. Expression: Class I antigens are expressed on all nucleated cells except those of the central nervous system and platelets. The class II antigens are expressed on antigen presenting cells such as B lymphocytes, dendritic cells, macrophages, monocytes, Langerhans cells, endothelial cells and thymic epithelial cells. Cytokines, especially interferon gamma, that is IFN gamma, increase the level of expression of class I and class II MHC molecules. Significance: haplotyping of MHC is necessary in certain diseases for tissue transplantation, etc. Thus, they play major role in graft rejection or acceptance. To summarize at the end, major histocompatibility gene family is divided into three subgroups: class I, class II and class III. MHC class I interacts with CD8 and class II interacts with CD4. MHC class I occurs on all nucleated cells apart from RBCs and presents epitopes to killer T cells, also called cytotoxic T lymphocytes or CTLs, and MHC class I helps mediate cellular immunity. MHC1 occurs as an alpha chain composed of three domains alpha 1, alpha 2 and alpha 3. MHC class II can be expressed by all cell types, but normally occurs only on professional antigen presenting cells, macrophages, beta cells and especially dendritic cells. These molecules mediate immunization. MHC class II is formed of two chains alpha and beta, each having two domains alpha 1, alpha 2 and beta 1, beta 2. Histocompatibility genes are inherited as a group, that is a haplotype, one from each parent. Thus, MHC genes are codominantly expressed in each individual. That's all about major histocompatibility complex. I end up my session here and I am sure all that we have discussed here will be helpful to you. Thank you.